everyone, my name is Katrina Shinadliklikan, and today I'll be demonstrating to you the internal and external structure of a pig's kidney. But first, I'll show you the different supplies that I'll need to dissect a pig's kidney. Now let's go! In addition, you need to wear gloves while conducting the procedure. After wearing gloves, we are now ready to conduct the dissection process. So let's go! So this is what we call ureter. Urine is transported from the renal pelvis into the bladder by the ureters, which are bilateral thin tube structures that join the kidneys and bladder. So this is what we call renal artery. Large volumes of blood are transported by them from the kidneys to the aorta, while the renal vein, blood veins that carry blood from the kidney back to the heart. And this is what we call the renal cortex, where the blood filtering units known as nephrons start. While this is what we call the renal medulla. The medulla's primary job is to control the urine's concentration. Renal pelvis. Urine is generated and collected in a huge chamber called the renal pelvis. Unfiltered blood is seen in the hyaluronic region of the kidney after entering through the renal arteries. It then splits off to form the segmental arteries and travels through the renal columns, which are the spaces between the renal pyramids. Thus, the term interlobar arteries refers to these blood vessels. Subsequently, it will divide into the arcuate arteries at the base and top of the renal pyramids. and then branch out into the interlobular arteries and cortical radiate arteries in the renal cortex. These blood vessels supply multiple nephrons, specifically the glomerulus, where blood filtration takes place. Afferent arterioles provide the glomerulus with blood flow. Observe how our filtered blood leaves the body and enters the afferent arterioles, diverges into the peritubular capillaries, which supply blood to the proximal portion of the cell. Convoluted tubule, the collecting ducts, the vasirectum, and the loop of Henle. From there, it converges once more into the cortical radiate veins or the interlobular veins, passes through the arcuate veins at the base of the pyramid, and returns blood to the renal columns and interlobar. It then travels to the renal veins and the hyaline region before being sent to the inferior vena cava of the heart. Now, after conducting the dissection process, we are now able to locate the internal and external structure of a pig's kidney. Not only that, we are also able to find out how or what is the process of blood flow in our kidney. I hope you've learned a lot, especially in terms of the internal and external structures and what are their functions. And that's all. Thank you.